say that we want to do non-chemical management, but there's a way to do it. It simply can't be swapping out product, as I mentioned last night. That's going to fail. So we're not a product-based system. We're a system that begins with the soil. And once you spend some time and you understand this and you work with these microbes in the soil, they in turn are going to work for you or work for us. So that's the message that you take when you communicate, back home and take these messages back home and you talk to park superintendents, you talk to decision makers, and trying to get them to understand exactly what it is that we're talking about. So it's very different because they've been trained differently. They've been taught differently. They've had a whole lot of the marketing coming back that you have to use these products. And if you don't use these products, it's simply not going to work. If you want to be successful, if any park district, school district, large property, small property, anywhere in the country wants to be successful, we absolutely have to adopt the approach and the practices that T and Andrea and Paul are going to talk to you about, because it just simply won't work. You know, the idea of the program where we've been taught to apply a chemical nitrogen. Nitrogen is the savior. Nitrogen fixes everything. You saw T's pictures last night. Nitrogen had very little to do with what that was, if anything at all. But that's a commodity. That's a product. There's vested interest in selling as much nitrogen as they can. So what we're talking about is not something that is wrapped into the commercial side of the industry. When you apply, Paul will talk about when you apply salt-based nitrogen, it's going to be disruptive to everything that you're going to see on that screen. It's going to mess it up. It's not going to allow it to work properly. So that's the first thing, has to go away. If we make a commitment to get rid of pesticides, to get rid of synthetic fertilizers, you absolutely have to make that commitment that all these salt-based synthetic things go away, because it is the organic approach, that those, it's just simply the breaking down of organic matter and getting a healthy nitrogen cycle happening down in there. So we're way beyond nutrients in a bag. Are organic fertilizers necessary at some point or part of a program? Certainly. Until and only until you get everything that they're going to talk about functioning. And then they become less important. So many of you have maybe heard that organic management or natural management becomes less expensive over time, because there's a transition period. There's a time to go from point A to point B. Once we get into that, and then most of the way through that, everything that they're going to talk to you begins to click and begins to start to work. And now we break the dependence upon purchased product, and that's even purchased organic product. So it comes down to more feeding these guys that you're going to see on that screen, as opposed to feeding the tree or the grass. And we're not feeding them anyway. There's no food whatsoever in a bag of fertilizer. That's all just raw materials that act as catalysts in physiological processes of the plant. So the concept that somebody's selling you plant food is just a marketing concept. So, do we need a little bit of supplemental nutrition on certain plants in the landscape in the beginning? Maybe, maybe not. But eventually we begin to, move in this direction. So in my work, I could not succeed. I couldn't be successful if I focused on the bag, box, or bottle. I have to focus here on everything. Doesn't matter whether it is a lawn, an appropriate area of grass around a residential property, or whether it is later on I'll show pictures in another workshop of a Division I soccer field out on the West Coast that is pristine and is naturally managed. So it all of that, big or small, depends upon this. So Paul Wagner is the president of Greener Pastures Organics, property care company located in Southampton, New York. He's also president of Soil Foodweb New York, a commercial soil testing laboratory in Center Moriches. They do all of the testing for me. I don't engage in any project unless, I send my sample first to Paul. He's a board-certified master arborist, as well as a New York State certified nursery professional.
All right, so where a lot of this really starts is kind of in a forested environment. This is where a lot of our, or in a natural environment, I should say. A meadow is kind of the ecosystem we're trying to mimic when we mimic a a lawn environment, when we're dealing with a landscape with a lot of woody plants, we're trying to mimic a forest environment. So, the general fertility program for a forest in this area of the country is the leaf litter that it drops, dead branches and things like that. So that's what naturally nurtures a soil health. So a forest drops about 4,000 pounds of leaves per acre each year. So that's a lot. When you take a tree out of the forest, trees in the landscape don't know that they're not in a forest. So they don't have all these biological associates, they don't have all the leaves, they don't have all that stuff. And so a lot of times they can get sick, they can get problems, and I just have one example up here, which is bronze birch borer. I'll get into the tax collector thing in a couple of minutes, but basically all plants pay taxes, just like you and I. You thought it was just you and I? All plants have to pay taxes as well. All these insects and diseases in the forest actually have very important roles. When you see a tree that, maybe was broke in a snowstorm, bronze birch borer is what's going to, if you think on the kind of Darwinian realm, bronze birch borer is there to put this tree out of its misery and to take whatever resources are left and redistribute them to the community, okay? So it's going to cycle all of that material back into the forest. So that's kind of the role of things like bronze birch borer, or I have an example coming up, which is Phytophthora. Things like that are not really pests in a forest. They're pests in a landscape. And the big difference is a forest has this whole ecosystem that keeps the trees healthy. It's only the ones that, maybe were damaged, whether it was from, a buck maybe rubbing and removing some bark, that a tree gets damaged and Mother Nature kind of sends in these insects and diseases to get rid of that damaged plant. So healthy plants, as they grow, the green tissue produces photosynthesis, like T was just speaking about. And all the energy that the plant makes through photosynthesis, on a healthy plant, up to, I should say, 80% of that energy goes into the root system. About half of it is for the growth of the roots. So if you think about a lot of the energy a plant makes goes into growing roots. But there's another investment that the plant makes that goes through the root system, and it's called exudates. There are things that the plant releases right out of its root system in order to feed microbes right around the root system. These exudates those are Mother Nature's taxes. So, almost half of all the energy that a plant makes, healthy plant, will be released out of the root system into the soil in the form of these exudates. And what they really are foods for bacteria and fungi. So in my CGI root over here, this is all bacteria, fungi that's feeding on these exudates, and they are actually protecting the root or making a shield around the root to protect it from things like disease. And so a tree in a forest that's very healthy produces a lot of photosynthate, and a lot of those exudates go in the root system, and they keep the plant healthy by growing all these beneficial microbes. When the top breaks out of that tree and the tree can't pay its taxes, so to speak, can't produce these exudates because it doesn't have enough leaves anymore to photosynthesize, the amount of exudate coming out is much more limited. The tree can't pay its taxes, and then Mother Nature sends in the tax collectors. Now, for another kind of cliche, this is where most of the traditional people come in and shoot the messenger with a pesticide. I'm going to take you through just one project, and I'm sure T has dealt with this quite a bit. I know Chip has, but this was a huge project that was done a lot of the trees had to be planted prior to construction on this construction site because there would be structure and other landscape installed after the fact. So this tree was planted in, and it's a beech tree. It was a fall dig, which was is kind of a no-no to begin with, but that was installed in November or late November, and then this is the following March, what we're looking at now. So you can see as the construction site kind of progressed, we're losing all that soil structure. T talked about having 50% of our soil solid and 50% pore space for air and water. In this case, we've lost all soil structure on the construction site due to compaction. There were some tree protection zones for the trees that were recently installed. That's a Zelkova. So this is a lot of what the site looked like. This is 5 months after that initial picture. So this is early August. That's the same tree. There's our beech tree with all this landscape around it, guest house here, tennis court there. So the site's changed quite a bit. But we have all these underlying soil problems now. Now this happens. We have Phytophthora popping up because, again, this tree doesn't know it's not in a forest. All those things that would have protected it from Phytophthora in the forest are not there. So now we get this great question. We now put a guest house is where our access was to plant this. So this is now going to cost us $100,000 to replace it if it dies. What can we spray to save it? And we start the cycle of chemical dependency in the landscape. And this is where starting out with a broad spectrum fungicide to kill the Phytophthora kills the entire soil environment. And then we have no nutrient cycling. We have none of the other benefits that all these microbes that would keep the plants healthy, they've all been killed off, and we're now on basically life support. So we do it a different way. We work on building the right soil biology to nurture the soil. That is essentially what's going to compete with the disease. And the question of, can we do this organic or not? That question has been answered a long, long time ago. It is possible to do anything that you can do with chemistry, you can do with biology. You can do it organically. So what is soil biology? Now, even though I'm up here, there was a point in my career where I would be the person with the spray gun trying to kill off bacteria, fungi, protozoa, nematodes. I am now the person who has all those things coming out of my spray gun as I'm trying to nurture all those beneficial microbes in the soil. So bacteria and fungi, what do? What are their functions? So they decompose organic matter. When you put leaf litter out, when you put compost out, when you put mulch down, you put organic fertilizer down, it's really, you're feeding bacteria and fungi. Those are the guys that are going to break that stuff down and retain all the elements that were in that organic matter. So if an organic fertilizer is put down, the reason in the organic system why we don't have leaching is because the bacteria and fungi decompose that fertilizer, they sequester the nitrogen, the phosphorus, the potassium, and then they stay in the soil, they don't leach. I'll get into the exact way they do that soon. So they retain all the nutrients. They also build soil structure. So going back to that project where soil structure had been completely destroyed, it's really the biology that's going to work on starting to rebuild that soil structure. This is a picture of a bacterium. And so the sphere in here, that's the body of the bacterium. There's these little wings on the side there. That's a glue layer that bacteria put around themselves to stick themselves to their food resource. They stick sand, silt, and clay particles together, And that's the beginning of production, or that's the beginning of really the building of soil structure, is that the bacteria will glue soil particles together and make aggregates. Okay, so here are our aggregates. You can see all the different colors and textures in there. So that's sand, silt, clay, and organic matter that the bacteria have glued together and created those aggregates. And now that you have aggregates, you also have all this pore space, again, trying to meet that 50% level where we can have some water, we can have some air. We want a highly aerobic environment, not compacted. And so the bacteria are the start of that. These strands are mostly fungi. Fungi will grow in long strands, and they wrap those microaggregates that the bacteria create, and they wrap them into macroaggregates, or larger aggregates. This is a corn leaf, a few weeks after it laid on the soil surface, and this is all fungi growing in these long strands, and they are slowly decomposing that corn leaf. And again, when you get rain or irrigation, these long strands of fungi that now have all the nitrogen that was in that corn leaf, they're not going anywhere when it rains. So that's, again, how the biology retains nutrients so that it doesn't leach, does not end up in our groundwater. And again, we hear a lot, well, how come you can use so much less product on the organic program than you can on a chemical program? And it's because we have a means to keep it in the soil if we are growing all these beneficial microbes. So they help us out. They're our source of nutrient retention. So 1 gram of typical forest soil in the rhizosphere, and the rhizosphere is basically the area right around that root zone. So if you just think about that root system that, or one of those CGI roots I had there, right around that, a gram or a teaspoonful of soil would have about 10 billion bacteria. And there would be in one little teaspoonful miles of hyphae, like you just saw on that corn leaf. You would see miles of these strands of hyphae growing in 1 teaspoonful of soil. Now, in a teaspoonful or gram of soil that's outside the rhizosphere, where those exudates aren't present, you'll still have a lot of bacteria, but you're down to about 600 million rather than 10 billion. So you just think about the difference. That's how powerful all those foods are that the plant is putting out to grow so many microbes to keep the plants healthy. So bacteria and fungi, again, they're decomposers. These are the ones that are going to, again, do the initial breakdown. You put leaf litter down or wood chips, they are going to break it down and kind of convert it into soil, so to speak. And there was a question earlier, are we trying to get the soil to the place it was 1,000 years ago? And what we are trying to do is mimic a natural environment. So this is forest duff, and you can see all the white mycelium, all the fungal hyphae growing in this bit of duff, which is, some broken down leaf litter, some root mass, some, woody materials, et cetera, on the forest floor. This is in a landscape we're managing where we've added granular humate, and then we've sprayed compost tea over it. And the fungi in the tea has colonized that food resource we put down so we can mimic that natural environment. We're trying to, again, rebuild that forest floor type of environment, but we're trying to do it in the landscape. So basically your plants, those are your primary producers. They go through photosynthesis. They make exudates. They make roots for organic matter. They make organic matter. They drop that in the soil. The exudates and organic matter feed bacteria and fungi, and the bacteria and fungi protect the plants from insect disease. They retain the nutrients, and they build soil structure. So it's all part of the system. Now, there are a couple of different ways that bacteria and fungi provide this disease suppression. Sometimes, and you may have seen this photo last night, but this is a nematode trapping fungus, again, which would grow in the root zone, right around the roots of the plant. And basically, the nematode would swim through one of these hoops, the hoop constricts, and then the fungus decomposes the nematode. It's a true dog-eat-dog world in the soil. So you have your disease suppression or pest suppression, pathogen suppression. Sometimes it's direct antagonistic behavior from one organism feeding on another. Sometimes it's just, like a sheer coat of so many microbes that the disease-causing fungus that's in the soil doesn't even know that the root is there. And so sometimes it's just a matter of outcompeting one critter or a group of critters outcompeting a disease or an insect. Sometimes it's one organism feeding on another one. So moving past bacteria and fungi, we get into predators. So these are things that feed on bacteria and fungi, and then all the nutrients that the bacteria and fungi held onto now will be cycled back to the plants. So you have protozoa. These are primarily things that feed on bacteria. They make nutrients plant available, and they also build soil structure, but they're going to be slightly different than bacteria created the little aggregates. These guys are highly mobile, and so they will move the aggregates around and have a little bit more active role in boosting your soil structure. This is a little video clip. So there's an amoeba on a microscope slide. These are flagellates swimming around. These are both two different microbes they're two different types of protozoa that feed on aerobic bacteria and then cycle the nutrients that the bacteria were holding on to back into plant available form. Then we get into nematodes. So where protozoa focused on bacteria as their main food resource, nematodes have many groups of different things that they feed on. So some nematodes feed on bacteria, some feed on fungi, some feed on plants. Those are your root feeders, those are your bad guys. There are then insect pathogenic nematodes. Those are really, really good beneficial guys that will control insects, and there are nematodes that feed on other nematodes. These are very much like miniature earthworms or microscopic earthworms, similar to that. So they move quite a bit in the soil, and again, they have a very active role in building soil structure. So that's the head of a bacterial feeding nematode. That's an acroboles, in case you wanted to know. I know you spend your life in a laboratory, these are the things that get you excited. This is a predatory nematode. And T kind of specifically mentioned this in regard to soil structure. You want to have a lot of pore space in your soil, because if you compact your soil, this root feeder, you can see how much smaller he is than the predatory nematode. You compact your soil, you may choose for this guy to survive, but this guy not to be able to access his food resource. And so that's our main way to control root-feeding nematodes, is through predatory nematodes. So thinking about, again, this is a little bit of where the rubber hits the road when people tell you, well, the chemical system is it's much easier, you know what you're going to get all the time. The bottom line is there's a lot of, in a healthy soil, you have a tremendous reservoir of nitrogen sitting out in your soil at any given time. This doesn't include all the microarthropods. This is just bacteria, fungi, protozoa, and nematodes. In a general healthy landscape, you have about 10 to 15 pounds of nitrogen just as a reservoir that's in the soil at any given time. Your protozoa and nematodes can cycle 2.5 pounds of N per 1,000 square feet per month in a healthy soil. So what does that actually mean? So in an area like New York City, where we are now, we would have frozen soil maybe for 2 months of the year, maybe not so much this winter, but in your average year. So you figure over 10 months where the soil's not frozen, 2.5 pounds of nitrogen per month per 1,000 square feet cycled. Now, how does that compare to a fertility program? If you look at some of the chemical lawn guys, they usually put down 3 to 5 pounds of nitrogen per 1,000 square feet per year. But I would say, if you just take care of your biology, we have 5 times that already present that we can access and make plant available. So you can add your chemistry, or you can manipulate your biology and make that plant available. If you looked at the packaging on a general tree fertilizer, they usually will be putting down nitrogen at the rate of about 0.5 pound to 1 pound of nitrogen per 1,000 square feet. So you have 25 to 50 times that available of what you would be applying in the chemical realm. You already have it in your soil. It's just a matter of making sure you're taking care of your biology and tapping into it. So it's all there. It's a matter of knowing how to make those things happen. So I'm going to go over just a very quick overview of building healthy soil. We need organic matter. That provides a great environment for the microbes to live. You need a diversity of different microbes, and every ecosystem is a little bit different. So in a forested system or a tree system, you want to have a fungal-dominated soil. So you want to have maybe 10 times to 100 times more fungi than bacteria. But in a grassland or a meadow, you want to have about equal amounts of fungi and bacteria. So adding a diversity of microbes is great, because if we put 20,000 species out there in a compost or a compost tea, let the plant put their exudates out and select which ones they're going to feed, and let that environment kind of create itself. And we just have to put all the pieces in the right spot. And then we want to add some specific microbes if needed. Going back to that struggling beech tree that the client's asking now, which pesticide do we use to kill the Phytophthora? I say there is a pesticide we can use, but it's called root shield, and it's a beneficial fungus. And it's one fungus that will kill the Phytophthora in the soil. That's its job, and that's what keeps Phytophthora at bay in the forest. So basically, you can purchase this natural product, or you can create a great diversity of microbes in your soil and outcompete the Phytophthora through use of maybe some specific compost teas or some compost applications. But on occasion, some specific microbes are needed. And then you get to the point where you basically want to feed the soil microbes and then allow the soil to turn all those things over to the plant. So where do we get our organic matter? For starters, we could just leave the organic matter that your landscape creates. That would be the best way to do it. So love them and leave them. The leaves, again, going back to that initial slide, a forest is going to drop 4,000 pounds of leaves per acre. That's about 100 pounds of leaves per 1,000 square feet. Just think about that. You're removing that 100 pounds, you're not only removing all that, all that material, but think about all the critters that, rely on that food resource for food. And if you do have to take them off and put them back in a more palatable form, like compost or a granular humate or something, you want to make sure that you're feeding all the microbes that would generally be in that environment if it was in a forest or a meadow. And so whether you do that with boosting your organic matter, can be done through leaves, they can be done through wood chips. The reason that we would prefer a source like leaves is leaves can get mixed in the soil by earthworms for free. So anything that's pliable, like a leaf, where a wood chip is not very pliable, but a leaf, an earthworm, this is a corn leaf, everybody knows how large a corn leaf is, an earthworm has come up to the soil surface, grabbed it, and pulled it down in its burrow. So you're talking about free soil mixing. No more paying for tilling, no more any of that. These guys work for free, and they work 24/7, and some of the byproducts are, you have your aeration holes. These guys are aerating as they work. They're bringing organic matter down, so they're starting to build, like T said, we don't want to scrape off that A horizon, but why don't we let the earthworms start to build those horizons in the soil? We can add a little organic matter up top, and by the way, there is 10 times more available nitrogen in earthworm castings than in surrounding soil. And the reason for that is that there's 5 times more nitrogen-fixing bacteria, which pull nitrogen right out of our atmosphere and put it right in the soil. 5 times more in earthworm castings than surrounding soil. So when I look at that, I only see one thing: free fertilizer. If I see that on the soil surface, I know, you know what? I start thinking, instead of how much do I have to put down, how much can I take away? How much do I not have to add? And so then one other thing, you probably heard a lot about compost teas. Anybody brewing compost teas?
Once.
Okay. I know you just got a brewer you're working with. And so compost tea can be a very good tool. Again, there's no silver bullet. It's not like a fix-all. But I just want to take you through a couple of quick slides on the compost tea brewer. So here you have a bag with compost in it. There's aeration in the bag, aeration in the brewer. And you run the brewer. You can see the difference in that. Can everybody see the difference in that water? What you're actually seeing, that's just 1 minute. And think of it like one of those old percolator coffee pots. Very similar. They should have called it compost coffee instead of compost tea. But very similar process. You're pulling some of that particulate matter out of the compost, all the microbes out of the compost into solution. They multiply. And this is what happens. You have a tiny little piece of organic matter coming off the compost out into the tea. You have fungi latches onto it, starts to grow, and you will see this, by the end of the brew cycle, that may look like a road map. There'd be so much fungi. So you got your 200-gallon GOT brewer. This one, you're basically, every time you brew compost tea, you're getting the equivalent of 2 to 2.5 yards of compost out of that brewer. It's just in the form of liquid is the carrier. And then there are many things that you can buy over the counter that can help you achieve your goal. And I just wanted to mention, the trichoderma. Sometimes you may have to purchase something to deal with that Phytophthora while you're building up the rest of that soil. So there are some things that can help expedite your results. And this, again, is back at that site, but you can see over the course of 1 year where we're starting to build O horizon, A horizon, and there you can see the existing parent material soil. But over the course of 1 year to build, some soil, it's that's a pretty good process, and ultimately that's what will keep that healthy without having to reach for those pesticides.
That extract? Did you just extract the soil out?
That's a soil probe that we yeah.
And this is
This is a root feeding.
Oh, so maybe Cody?
Paul, can you explain quickly, just explain what you do when you do a food web analysis, and then we
So-
do that report
And how
that
would
you showed last night?
So that's a root feeder, that's a bad guy.
Yeah,
And you can see the stylet is short and robust, very well adapted to kind of jabbing into roots.
It's almost like a nail. You can see that.
So when somebody sends us a soil test, basically, and again, I'll just go back to trees as an example. I have a birch tree in my yard, it's got a problem. They send a soil test in. What we actually do is we go through a dilution. We, believe it or not, we count the amount of bacteria that are in that soil. We kind of dilute it to a level where we can accurately count. We go through a number of counts and then average so that we can get as close as possible. So we can say, hey, you have 400 parts per million bacteria. And then believe it or not, we would measure, if anybody looks through that microscope, there's a grid in the eyepiece. So we know, based on the field of vision, how many micrometers that grid represents, and we can
And that's how you do counts.
we measure the fungi, we count them, and so then we can say, well, you have 300 parts per million fungi, 400 parts per million bacteria, but we're in a tree soil, we really need this to be up around 3,000 parts per million fungi. Then what we do is we go through a dilution, and we do replicates for protozoa, and we allow them to kind of grow out, and we see how many protozoa you have in your soil, and that gives us an idea of how much nutrient cycling can you expect out of that soil. Once we know the amount of nutrient cycling, we can easily say, gee, this is providing us 100 pounds of nitrogen per acre. Well, you can easily say, all right, so I have roughly 50,000 square feet in an acre, 100 pounds of nitrogen. That's 2 pounds per 1,000 square feet. If I'm growing a fescue lawn, that may be plenty of nitrogen, I don't need to add any. But you can easily get to that number to see what kind of contributions do you have to make. And so we look at protozoa, we look at 3 different types of protozoa. Ciliates would tell us that your soil is anaerobic. Flagellates and amoeba would tell us it's highly aerobic.
You all saw a ciliate yesterday, moved around very nicely. So,
And then we want to warn you if we run across these guys.
Yeah, how do they eat that? Are they eating it like swallowing it? Like, is this like a?
It's,
It's
so,
not
all
like
right, so this is a predatory nematode right here. You can see there's a large cavity in the head. And so it actually would, just like a snake.
Yeah. So
Can you
it's
tell
not
us?
it's not gonna, it's
Can
not
you tell
gonna
us to
kill
get
it
down?
like that. It's not
No,
gonna.
what happens is if you get a close-up, and I could just bring a picture up also. I will do that now, just so you have another reference, but there's a tooth inside the head.
Oh my gosh. My son would love this.
You passed it.
So, can you see the tooth up top?
Yep.
That holds it in place, pierces basically the exoskeleton, it's not really an exoskeleton, but the epidermis of the nematode, and it actually just sucks out the cytoplasm of one nematode does the other.
And then his.
It's not going to chew it like, it's not going to eat it like a snake, it just pierces and then
sucks it.
And then,
Exactly.
And then there's gonna be a skeleton, or like the outside remains in the soil, or?
Right, it's just bacteria will be trying to get their bit of nitrogen out of that as well.
Okay.
and then explain this concept of inactive versus active.
Okay, so then what we look at is the amount of bacteria and fungi that are in your soil. There's a overall amount, like there's 20 people in this room and one person fell asleep, which is a very low percentage usually in a biology class.
I think half the people here are asleep though. Paul.
So that would be 5% are inactive, 95% are active. And that every organism has a seasonal fluctuation, just like your trees, some of them have leaves during the season and they drop leaves during the winter. Same thing with your microbes. There are some that are highly active in the spring and summer. Their activity drops in the fall. So the test would tell you if you have only 10% of your bacteria and fungi are active on April 15th in this area of the country, something's wrong. You may have a limiting factor. That could be your soil is too dry, and if we're having ample rainfall, too dry a soil means you don't have good soil structure. You don't have that 50% to hold the moisture. So, or maybe it was killed off from a pesticide. That's a very common, when we're on a golf course, a lot of times, or somewhere where it's heavily managed, a lot of times we're saying, gee, it's May, and you guys are, yeah, I know the grass is growing. There's a reason why the biology's not active. And it's probably because it was a very harsh chemical applied or something like that, and they all went dormant as a defense mechanism, or some of them died and some of them went dormant. So we look at active versus total amount and, plot that against what the rest of the, all our database was initially developed from, meadows are the kind of blueprint for grasses, and forests are the blueprint, because again, The tree on your lawn doesn't know it's not in a forest. So you want to create that same forest environment just in your lawn, in your front yard or wherever.
If somebody got a soil sample and sent it to you, they have a diseased tree, could you look at that soil sample and see what kind of an additive they need?
We wouldn't be able to do pathogen testing, but there are many labs that do that.
Yeah.
And that would be probably more pertinent. And the thing that I would say, one of the things that I do, and T's been on this, T had Battery Park City organic before it was the chic thing to do. And the thing is, there's almost always a way to look at a life cycle of a disease or insect and figure out where can I interrupt that without reaching for a chemical pesticide. And it's, if you're dealing with lace bugs, it's very simple. There's a time where you can spray vegetable oil, clog the breathing pores, and you can get excellent control. With leaf miner, there's a beneficial insect that feeds on leaf miner and boxwoods. We're working with boxwood blight now on one beneficial fungus that kills the blight, that attacks the blight.
What about borers, the ones that go into the...
So any insect like that, where you have a soft-bodied insect that you can get beneficial nematodes into one of the body cavities, I don't care which one, you can control that. Whether it's gypsy moth in the top of a tree, they've done all that research. I mean, that was in like 1995. They did that research. Beneficial nematodes will get, if you can get it to the insect, you can control it.